Welcome back to Module 6 HIV/AIDS. This is Dr. Aditi, Assistant Professor, Department of Chemistry, St. Joseph's College for Women Autonomous, Visakhapatnam. In this chapter 6.2, we'll be learning about immunity CD4 cells and CD8 cells. Chapter 6.2.1 deals with the description of CD4 cells and its action. 6.2.2 deals with description of CD8 cells and its action. 6.2.3 deals with the difference between CD4 and CD8 cells and their similarities. The outcomes of this chapter are to understand the importance of CD4 and CD8 cells for the immunity, differences and similarities of CD4 and CD8 cells. CD4 cells. CD4 cells means clusters of differentiation 4. They are glycoproteins present on the immune cell surface such as monocytes, T helper cells, macrophages, and dendritic cells. CD4 T cells are pivotal, playing a central role in immune protection. White blood cells are an indispensable part of the body's immune system. A type of white blood cells, that is CD4 cells to be specific, are infected and destroyed by the HIV, that is nothing but human immunodeficiency virus. With the loss of these CD4 cells, in such cases, the immune system and its responses decline, turning incapable to fight and attack pathogens. When the large amount of CD4 cells are lost, HIV-infected people are at a risk of developing AIDS-related opportunistic infections. What are CD4 T helper cells and why are they important? CD4 T helper cells are a type of white blood cells called lymphocytes that serve an important role in immune system. These cells help defend the body against infection by directing other killer cells to neutralize foreign agents. Without CD4 T helper cells, the immune system cannot recognize and coordinate a defense response to diseases like HIV. When these cells are significantly depleted due to the disease or other causes, the immune system can become blind to foreign agents and unable to defend itself. CD4 T cells are deemed to be helper cells as they stimulate the immune system to respond to infections rather than neutralizing infections. Responding to this CD4, the CD8 T cells act on them as they are considered as killer cells. They do so, that means the CD8 T cells do so through the production of antibodies helping to fight against foreign particles or pathogens. For HIV to be able to infect host cells, it has to multiply. For this, CD4 cells are its prime target. HIV attaches to these cells during infections, unloading its genetic content so as to alter the genetic coding of the host to finally generate HIV virions. In this process, the CD4 cells of the host are destroyed. Consequently, the capability to stimulate immune responses to defend in such people declines and eventually reaching a point where the body is susceptible to opportunistic infections. These are the CD4 cells and these are the virus which are infecting these CD4 cells. CD4 cells protects us or protects the immune system against infections and illness. It's important for all people with HIV to have regular blood tests. The two most important blood tests are for CD4 and viral load. The CD4 count tells you how many CD4 cells there are in the drop of a blood. The more they are, the better. Viral load measures how much HIV is there in the drop of blood. You want to have as little HIV as possible. During the initial acute stage of HIV, CD4 T cells can bring the infection under the control because their numbers are high, but they don't fully clear the infection. Instead, the virus will go into the hiding, embedding itself in tissues where it can remain undetected by the immune system for years. Here, if you have observed, the CD4 cell count is more and the viral load is very less. So here as the CD4 count is more, the virus hides itself. So it is very impossible or very less chances to detect the virus at this stage. In the meantime, the viruses remaining in the circulation will turn the tables on the immune system by binding it to the CD4 T cells and inserting their genetic material in them. This effectively reprograms the infected cell, turning it into an HIV producing. After producing thousands of new viruses, the infected cell eventually dies. Over the course of years, as more and more CD4 T cells are killed, the immune system gradually loses its ability to fight off and otherwise harmless infections. These so-called opportunistic infections are what account for the majority of illness in people with the advanced HIV. In this picture, you can observe the CD4 cell count is less and the viral load is more and more. As the CD4 count is less, this situation is not at all good. Role of CD4 T cells in HIV treatment. In the past, treatment for HIV was delayed until the CD4 dropped to a certain level. This was due in part to the toxicity of early antiretroviral drugs as well as the speed at which the drug resistance developed. Today, treatment of HIV starts immediately upon diagnosis. Antiretroviral drugs work by blocking stages in the virus life cycle, preventing the virus from making copies of itself. The drugs don't kill the virus but reduce its numbers to undetectable levels where it can do the body little harm. As such, the CD4 count plays no role in the initiation of treatment or the selection of drugs, but best suited for specific HIV type. The result of treatment should be the CD4 going up and the viral load going right down. That means by using or taking the drugs, what happens? The CD4 count is becoming higher so that the viral load will be lesser. These medicines are not acting on the virus, they help in increasing the CD4 count. This is how the medicines help to get the CD4 count higher. This is a picture showing how the HIV virus infects the CD4 T cell. This is the HIV virus and this is the CD4 cell. In the first step, what happens? Receptor binding to the HIV virus. The virus binds with a CD4 receptor. It just binds. In the second step, what happens? Membrane fusion occurs. That means the fusion of the membrane of HIV as well as the CD4 cell takes place. That means both combine together. In the third step, HIV DNA enters the cell. The HIV DNA, whatever is present, is released into the CD4 cell. In the fourth step, what happens? The HIV DNA combines with the human DNA. And from here, what happens? Infection becomes more and more. Inside the cell, several interactions occur between the viral DNA and cell proteins, allowing the DNA into the cell nucleus. This is about CD8, how CD8 is acting and CD4 is acting. CD4 helps in order to reduce the virus. But what about CD8 cells? CD8 cells kills the virus. This is the difference between CD4 and CD8. CD4 cells suppress the viral load, but coming to CD8 cells, they just kill the virus. CD4 cells are surface glycoproteins participating in activation and recognition of T cell antigens. CD4 helper T cells are a type of white blood cells that coordinates the immune response. It can help fight many infections but is ironically the target of HIV infection. As HIV progressively depletes these cells, the body becomes less able to defend itself against otherwise harmless infections. The CD4 count is a blood test used to measure CD4 T cells and the relative strength of our immune system. With early treatment, a person with HIV may return to a normal CD4 count of 500 or above. If left untreated, HIV will almost invariably progress to AIDS. CD8 plus, otherwise known as killer, they are cytotoxic, and CD4 plus are helper T cells. Killer and helper are the functional names of the cell surface protein CD8 and CD4. CD8 plus T cells, also known as killer T cells, are cytotoxic. This means that they are able to directly kill virus infected cells as well as the cancer cells. CD8 plus T cells use small signaling proteins known as cytokines to recruit other types of cells when mounting an immune response. A different population of T cells, the CD4 T cells, function as helper cells. Unlike CD8 plus killer T cells, the CD4 plus helper T cells function by further activating memory B cells and cytotoxic T cells, which leads to larger immune response. This is a picture showing the CD8 cell killing the cell which is infected by a virus. That means if a cell is infected by a particular virus, this CD8 cell kills that particular infected cell. Here MHC means major histocompatibility complex. Mostly CD8 cells work on this major histocompatibility complex. Here this is a picture showing how the CD8 plus T cells kills the infected cells. Any of the infections, the CD8 plus T cells kills those particular infected cells. CD8 plus T cells are important in the control of HIV replication, and these are the evidences. Emergence of HIV specific CD8 plus T cells coinciding with the decrease of viremia in acutely infected patients. A potent HIV specific CD8 plus T cell response to contribute to the reduction of the pool of HIV infected cells and the HIV reservoir. Increase in viral load in HIV infected macaques after CD8 plus T cell depletion. Associations between the frequency or functional capacity of HIV specific CD8 plus T cells and limited viral duplication or decrease in the non-progression of HIV infected patients. Appearance of viral escape mutations to evade the immune pressure of HIV specific CD8 plus T cells. Requirement of CD8 plus T cells for maintaining therapy induced viral suppression in SIV infected macaques. In the previous slide, macaque is nothing but a monkey. Now coming to the similarities between CD4 and CD8 T cells. CD4 and CD8 T cells are two types of T lymphocytes in the blood. Both contain T cell receptors whose variation helps to identify the help of T cells. Both differentiate from the common lymphoid progenitor in the bone marrow and mature in the thymus. Moreover, they are involved in triggering a cell-mediated immune response. Now let us discuss CD4 T cells versus CD8 T cells. CD4 T cells are the T cells that express CD4 glycoprotein on the cell membrane. CD8 T cells, the T cells that express CD8 glycoprotein on the cell membrane. Coming to CD4, they are also known as helper T cells. Coming to CD8, they are known as cytotoxic T cells or killer cells. CD4 T cells, here CD4 glycoprotein serves as the T cell receptor. Coming to CD8 T cells, CD8 glycoprotein serves as the T cell receptor. Coming to CD4, they recognize antigens on the surface of antigen presenting cells. CD8 T cells recognize antigens on the surface of all nucleated cells. CD4 T cells recognize antigens present along with the major histocompatibility complex class II molecules, and coming to CD8, they recognize antigens presented along with the major histocompatibility complex class I molecule. CD4 T cells induce other immune cells to trigger an immune response by secreting cytokines upon recognizing antigens. CD8 T cells induce cell death of the virus cell infected cells, tumor cells, either by cell lysis or apoptosis. Then what is the difference between CD4 and CD8 T cells? In the definition, CD4 T cells refer to T cells that express CD4 glycoprotein on the cell membrane. CD8 T cells refer to T cells that express CD8 glycoprotein on the cell membrane. CD4 is also known as helper cells. CD8 is also known as cytotoxic T cells. What is the type of T cell receptor present in CD4 and CD8? CD4 glycoprotein serves as the T cell receptor in CD4, while CD8 glycoprotein serves as the T cell receptor in CD8 cells. This is a picture showing how the CD4 and CD8 cells work in the human body. This is the antigen presenting immune cell, and this is the MHC2, that is major histocompatibility 2 class 2, and this is a CD4 T helper cell, and how it is being injected into the antigen and cytokines are released. When a CD4 helper T cell binds with a MCH2 antigen complex on an antigen presenting cell, both the antigen presenting cell and the T cell release cytokines. In the second step, what happens? In response to the cytokines, the T cell clones itself. The cloned T cells producing different cytokines that activate B cells and CD8 cells. Here, if you have observed this, the CD4 cells and the antigen combine together and cloning takes place where more and more T cells are produced, and finally CD8 is produced. Next, coming to CD8 T cells, when a cytotoxic T cell interacts with the MHC1 epitope complex on an infected cell, it produces granzymes and perforins. The perforins form pores in the plasma membrane. Granzymes enter the cell and break down the proteins lysing the cell. That means when cytotoxic 8, that is CD8 cell, acts upon the MHC complex of infected cell, what happens? It performs or it progresses into the infected cell and directly kills the bacteria or kills the infected cell. This is the difference between CD4 and CD8 in antigen presentation. Another difference between CD4 and CD8 T cells is that CD4 T cells recognize antigens on the surface of antigen presenting cells, while CD8 T cells recognize antigens on the surface of nucleated cells. Coming to MHC complex, CD4 T cells recognize the antigens presented along the MHC class 2 molecules, while CD8 T cells recognize antigens represented along with the MHC class 1 molecules. There is also a difference between CD4 and CD8 cells based on their function and role. CD4 T cells induce other immune cells to trigger an immune response by secreting cytokines upon recognizing antigens, while CD8 T cells induce cell death of the virus infected cells or tumor cells either by cell lysis or apoptosis. Coming to the conclusion, CD4 T cells are T helper cells with CD4 glycoprotein on the cell surface as the T cell receptor. They recognize antigens presented along with the MHC class 2 molecules by the antigen presenting cells. Then they secrete cytokines to induce other cells in the immune system, including B cells, CD8 T cells, and macrophages to trigger an immune response. On the other hand, what happens? CD8 T cells are cytotoxic T cells which express CD8 glycoprotein as the T cell receptor. They recognize antigens presented along with the MHC class 1 molecules on the surface of all nucleated cells and are responsible for the cell death in virus infected cells and tumor cells. Therefore, the main difference between CD4 and CD8 cells is the type of T cell receptor on the cell membrane and the type of immune response they generate. This is a comic play of CD8 cells, how it kills the viruses. See here you can see the CD4 T cell. It just identifies that some virus have been introduced into the body, and it thinks where to report now. Meanwhile, CD8 cells, what they do, they just attack the virus and kill the virus. When does this CD8 cells kill the virus? When more and more CD4 cells are dying, then CD8 cells come into action and kill the virus. Here you can see the CD8 are fighting them off, but they are not enough because CD4 cells, more and more CD4 cells are being dying. More and more CD4 cells are getting infected and dying. The HIV are replicating inside them. So when the HIV are replicating in them, what happens? Other diseases like TB, Candida, influenza, and other opportunistic microbes are taking advantage of the situation and are joining in the fray. So we need more and more CD4 T cells to be incorporated into that particular person where the HIV is infected. Here, this is how the CD8 cells finally kills all the virus and it clears the viral infection. Here you can see that finally only one cell is left and CD8 cell is ready to kill this, and finally no virus are left. In order to improve the CD4 count, there are some food habits to be induced into the body system. So broccoli, carrot, pomegranate, all the nutrient rich foods or vitamin C rich foods are being taken by the human body in order to increase the CD4 count. The structure and virology of HIV will be dealt in the next chapter. Thank you.